The lecture for Lab 7 on enzymes. Enzymes are molecules that you find in cells, and they're essential to life. Every living cell has to have functional enzymes to catalyze different metabolic processes. And these enzymes are catalysts. Sometimes we refer to them as biological catalysts. And a catalyst is something that increases the rate of a chemical reaction. It makes the reaction more likely to take place. And enzymes, as they catalyze chemical reactions, they will not be changed or consumed in the process. So they facilitate a chemical reaction, but they are not directly changed or altered by that reaction. And enzymes are highly specific in terms of each enzyme will bind only one type of substrate, and on that substrate they will catalyze only one type of reaction. So every cell has hundreds and hundreds of different enzymes, and each enzyme has a specific substrate and a specific type of reaction that it produces. And the vast majority of enzymes in cells are proteins. Enzymes have an effect on activation energy. And activation energy is the amount of energy that has to go into a reaction to get it to go forward. Enzymes increase the rate of a chemical reaction by decreasing the activation energy. So they reduce the amount of energy needed for a reaction to take place. And this is just a diagram of a reaction. So here are the reactants. The reactants are what you start with in a reaction, and those reactants will be changed to products. So in order to convert these reactants to products, you need to put in a certain amount of energy. So you have to boost these reactants up to a certain level so they can be converted into products. What enzymes do is they lower the amount of energy that has to be put into the reaction to convert the reactants into products. When talking about enzymes, there are two important terms that you need to know. The first term is substrate, and the substrate is defined as the substance the enzyme acts on. The second term is active site, and the active site is defined as the small part of the enzyme that binds to the substrate. So when you draw a picture of an enzyme, this is the enzyme. Every enzyme will have an active site, and the active site has a particular shape which will fit exactly the shape of the substrate. And sometimes we refer to the substrate as the reactant. But this is what makes the enzyme highly specific. Because of the three-dimensional shape of the enzyme can only bind to a substrate that fits perfectly into the active site. As I mentioned before, most enzymes are proteins, and an enzyme's specificity is determined by its structure. So it's very important for you to review the different levels of protein structure. And if you remember when we've talked about protein structure before, there are 4 levels of structure: primary, secondary, tertiary, and quaternary. And primary structure is the order of the amino acids, how they are covalently bonded together. And remember that there are 20 different types of amino acids, so the order is important. Then secondary structure. Secondary structure is how that original chain of amino acids starts to bind together via hydrogen bonds. So it can either be in an alpha helix shape, like a corkscrew shape, or it can be in a beta pleated sheet. So sort of like paper that has been folded like a fan. But both of these are held together by hydrogen bonds. Then the tertiary structure, this is the three-dimensional structure. This is how the whole thing folds in on itself to form that three-dimensional structure. And you can see alpha helices and beta pleated sheets in it. And this is also held by different types of bonds and can include hydrogen bonds. Then quaternary structure. This is how the one polypeptide binds with other polypeptides to form a functional structure like an enzyme. So for the protein structure, hydrogen bonds are very important for holding the three-dimensional structure of a protein, of an enzyme. The enzyme that you will be working with in lab is called invertase. And you know that this is the name of an enzyme because it ends in ase. All enzyme names end in ase. So this is the enzyme here, and sometimes it is referred to sucrase, also again the name of an enzyme. But here's your enzyme, and the active site is this site here. The substrate that invertase works on is sucrose. And sucrose is a sugar. A lot of sugars end in ose. And sucrose is a disaccharide. It is composed of two monosaccharides covalently bonded together. So when sucrose is present with an active enzyme, the enzyme will bind to the sucrose. It will bind to the substrate at the active site, and it will catalyze the reaction of breaking that bond. So then it is converting the substrate or the reactants into the products, and those products are the two monosaccharides, glucose and fructose. And then the enzyme, of course, is not changed in any way during the reaction. So once it is done catalyzing the reaction, it is ready to go again. So if there is more sucrose present, then the process will repeat itself. Any factor that has an effect on enzyme activity can be referred to as an enzyme activity effector. So something that has an effect on the enzyme's activity. And there are three main enzyme activity effectors. The first is temperature. So changing temperature can have an effect on hydrogen bonding, which will affect the protein folding for the enzyme. The second is pH. Changing the pH of the solution, and remember pH is a relative measure of the concentration of hydrogen ions in a water solution. This can have an effect on hydrogen bonding in the protein structure, which will have an effect on the folding. And the last thing that can have an effect on enzyme activity is concentration. So the concentration of the enzyme or the concentration of the substrate. So going through some of the enzyme activity effectors, the first one, temperature. So this is a graph that shows you how enzymes function at different temperatures. So on this graph, the x-axis is temperature in Celsius degrees, and the y-axis is enzyme activity. So how much product is produced as the enzyme converts the reactants to products. And if you notice in the curve, the high point of the curve, this is what we call the optimal temperature. So it's the temperature where the enzyme works at its fastest rate and produces the most product. And it looks like the optimal temperature could be about 37 degrees Celsius, which is body temperature. So remember, we are measuring in Celsius. So this is the optimal temperature. As you increase temperature, the activity of the enzyme decreases. And this is because you are denaturing the enzyme. You are having an effect on the hydrogen bonds, and so the enzyme loses its shape. And if the enzyme loses its shape, it loses its function. Also, as you decrease temperature away from the optimal temperature, the activity is going to gradually decrease. A second factor is pH. So pH, again, the x-axis is the pH scale, changing pH, and the y-axis is the enzyme activity. And again, you have a curve, and the top of the curve is the optimal pH. So this is where the enzyme works at its fastest rate. And this optimal pH looks to be about maybe close to 7. And most living cells prefer a neutral pH. Remember, 7 was neutral. And again, similar to temperature, as you increase pH away from the optimal pH, you get a decrease in enzyme activity. And as you decrease pH, you also get a decrease in enzyme activity. And that's because changing the concentration of hydrogen ions will have an effect on the hydrogen bonding, which again relates to the structure of the enzyme. If you alter the structure of the enzyme, then the enzyme can't function. The last factor is concentration, and this is an example of substrate concentration. So now the x-axis is increasing substrate concentration, and of course the y-axis is enzyme activity. So as you increase substrate concentration, you're going to get a steep increase in the amount of product produced until you reach a saturation point. So the saturation point, basically it's the point at which the enzyme is working as fast as it can. So even though you add more substrate, you cannot get a faster production of product. So the enzyme has reached its maximum rate as fast as it can go. Again, in lab, you will be working with invertase, that is the enzyme, and you will be determining if it can take the substrate sucrose and break it down to form the products glucose and fructose. Now, in order to be able to determine if the enzyme is functioning or not, you need to be able to detect the presence of glucose and fructose. And because these molecules are too small to be seen with the naked eye and too small to be seen with a microscope, we need to use an indicator, which will change color when these monosaccharides are present. If you remember from the organic molecules lab, the Benedict's test was used to determine the presence of monosaccharides. So the Benedict's test, you use Benedict's reagent, you add it to the solution, you boil it for 3 minutes, and then you will get a color change. And remember, this can give you a relative concentration of the amount of sugar. So if the solution remains blue, that means there is no sugar present. If it starts to turn green, that means you have very low levels of the sugar, the monosaccharide. If it moves towards orange, it changes orange, it's moderate levels. And if it turns red, then that means there are large amounts, higher concentration of glucose present. So as the solution turns towards the red color, that means the enzyme is working and converting more sucrose, breaking it down into glucose and fructose.